suppongo. Okay, so let's start with the next lectures, which are today they're more biological. So that's okay. So this idea you already have that this is kind of the central dogma of molecular biology, where you have DNA. DNA is translated into RNA, and RNA is transcribed into RNA, and RNA is translated into protein. And there are a few enzymes that are important here. This translation is the ribosome. This one, the RNA polymerase, the DNA polymerase. And the general flow is in this direction. So you have DNA is replicated, and then from time to time it is, or when you need to express protein, you express RNA and RNA to protein. So this is the standard flow. But there are alternative flows that are somehow special. So for instance, some viruses translate RNA to DNA, and there are RNA viruses, and sometimes it is translated into DNA and it is integrated into the host DNA, and there are many funny paths. Then RNA, there is a whole life sustained by RNA alone. So there is not everything is stored at the level of DNA, but some lower life forms, or viruses, they stay at the level of RNA. And it seems that the origin of life was more related to RNA, and then somehow it was found how to store the information more efficiently in DNA rather than RNA that is more unstable. And then in labs, it has been found a way to translate directly DNA to protein. But the central, so the standard flow is replication of DNA, and then transcription to RNA, and translation to protein. So yeah, I have a little bit of close-up here. So we have the DNA, then it is a double strand, so you have to open the two strands. You have to open the two strands. One of them will serve as a holding, as the template strand. So the strand is the one that is actually interacting with the RNA polymerase. But because of the complementarity, what you are expressing here is the translation of this folding strand. Okay, so you are acting on this, but because you have to copy the complementarity of each one of the bases, what you are actually is copying. And then in triples, we have three bases. This is called a codon. Each codon is translated by the ribosome into a polypeptide chain, and that is the protein, the final protein. So this is the standard theory. So this you already knew, and these things are less known. So first, there are regions that are not transcribed, so or that are not translated into proteins. And so, for instance, in this drawing, you have the green parts are translated into protein. Those are called exons, but there is a region that is not translated, and it's called an intron. And these introns, the lengths of these introns can be quite varied. So you see, this is a small intron, a large intron, and then even in and some regions can be shared by two proteins. So for instance, this region here is used to translate into a protein, but the same region is used to translate into a different protein. And depending on the path that you are, or the particular pattern that you have of translation, you may have all these names: convergent, divergent. So convergent means you start translating this one, and there is a region here that is shared. And Also, the direction of translation is not always the same. So when you are translated here, we are translating in this direction, but you could translate in the other direction. So it is pretty free. You can nest a protein inside the intron of another protein. So it can be really, really complicated patterns. And here you have an article that describes all these patterns. Then you have mutations. So, for instance, if you have a virus that has a genome, but this genome is not unique. So every copy of the virus, when making the translation, you have some mutations. So in your body, when you are infected, you have millions of copies of the virus. So each copy is different from each other copy, or in general may have some mutation. And then you put names to a few of them that are quite common. So for instance, you have this is Newcastle disease virus, whatever. So Newcastle disease virus, this is kind of the reference genome. And then this sequence that is relatively common. So it has these two mutations here in this region. So you put names to common appearances of these mutations, but every copy of the virus in principle could be mutated and is in general mutated. And then you have a phylogenetic tree of how the different viruses or how the different individuals of some species, how they are related. And with viruses and bacteria, because they change so frequently, they are very useful and they can diverge quite a lot. So you see, this is the phylogenetic tree of SARS and SARS-CoV-2. I don't know what it is, but I think it is all these ones here. I don't know what. This is SARS-CoV-1, SARS-CoV-2 here again. So you can see there is an ancestor genome, and then this ancestor genome is specialized into different species. So this is SARS-BAT coronavirus, this is SARS-human coronavirus, and different species. The virus specializes in that species, and inside that species it can diverge. And then here you have so the genome is different. How much is that translated into the different proteins of the virus? So here you have, for instance, 4 proteins, and then how many mutations are accumulated with respect to a reference genome.
What are the triangles on the tree?
I don't know exactly what they represent, what they want to represent here. I guess it is that there is some spread there, so it is not just a unique genome. But you would have to go into the article to see exactly what they mean by the triangles. Then DNA, we don't have a single source of DNA. We have 2 sources of DNA. One is nuclear DNA, that is kind of the human genome. But also we have mitochondrial DNA. So mitochondrial DNA is much smaller. So this is 16,000 base pairs. This is 3,000 million base pairs. This one is linear. This is circular. This one has histones. This one doesn't have histones. Here, the DNA is coming from your father and your mother. This one is only coming from your mother. So there are a lot of differences. And also it is the mitochondrial DNA is inside the mitochondria. It is replicated on its own way, so separated from the nuclear DNA. Then this we have already seen. This alternative splicing. So there are regions that are not translated into protein. So you have this region that is translated. So everything is translated into RNA, but not everything is translated into protein. So this region here is not translated into this protein. So this protein, this RNA has 5 exons, and then this RNA is using, or this protein is using the 5 exons, but this protein here is not using the exon 3, for instance. And then this is a protein, this is a different protein. And it is coming from the same gene, but the same gene can be spliced into different versions of that protein. So they are kind of related proteins because they share domains, but they are not exactly the same protein. And this happens typically in different tissues. So the same gene in a muscle cell is expressed like this, and in a lipid cell, it is expressed like this. And then there are also DNA is mobile. So mobile means that you have a sequence of DNA, and then there are mechanisms, and these are natural mechanisms. They happen all the time, and in living cells. So it is not just a molecular biology tool, but it occurs in real life. So pieces of the DNA, they are excised from the original sequence. Then the two ends are paired. So these two are gathered here. And then this extra piece of DNA is placed somewhere else. Actually, you may have heard of CRISPR-Cas. CRISPR-Cas was one of these mechanisms to take pieces of DNA from one place to the other. And this is called, there are several mechanisms. So this DNA transposon, LTR retrotransposon. Retro means RNA is translated into DNA and then integrated into the DNA. And non-LTR retrotransposon. Actually, all the sexual cells, so sperms and oocytes, in the generation of them, there is a lot of all these movements and rearrangement of genes. Then you have the DNA, but there is some control of the transcription, and this control of the transcription is there are several ways you can control, and one of them is through the histones. So you have to open the double strand of the DNA, so you can put somehow some marks in the histones to block or to favor those openings of strands. And the two most common marks are these histone acetylation, histone methylation, but you can also methylate the DNA. You can have this long non-coding RNA that somehow also regulates the translation because it interacts with transcription factors that we will see later. But you need to translate transcribe the DNA into RNA. And also you have these microRNA that are also somehow controlling the translation by the ribosome. And here you have this idea of microRNAs. So microRNAs, so let's say that there are small pieces of RNA that somehow binds to the messenger RNA, and then when it binds, somehow it is a mark to send the mRNA to degradation, and then how much protein is expressed in each one of the cells and each one of the conditions somehow is also regulated by the microRNA. So there is an equilibrium of how much microRNA is expressed, how much messenger RNA is expressed, and somehow you can control the final protein levels in very fine details. And the two proteins that are very well known to act here, they are these RISC and DICER. They are very famous proteins. And you can classify the genes in different categories depending on three. Well, you can classify in many different ways, but there are very well three that are very well accepted. One is molecular function, the other one is cellular component, where it is found, the product of that, and biological process. So these you can construct a tree of the different labels. So for instance, you have molecular function, and then you have many molecular functions. You have a million molecular functions. One of those molecular functions is ATP-dependent activity. And inside ATP, you have ATP hydrolysis activity. And you see, I haven't expanded all of them, but you see that you have a million molecular functions somehow categorized into a tree. And these three trees, they are gathered into this gene ontology. So when you want to say what a gene is doing, you have to describe it in terms of well-accepted labels, and this is one of the best dictionaries of well-accepted labels. And there are other ontologies, it is not the only one. Sequence ontology, protein ontology, pathway ontology. So each one of them, they have a focus onto a different topic. Then, when you describe a protein, then, for instance, this protein that we have it in the surface of our cells, and then you have here molecular functions. So molecular functions, you have this one, all molecular functions. So we have some molecular functions, GTPase activity, transferase activity, and the color, the intensity of the color means how many labels you have in this family. And then you have all biological processes. And if you go, this UniProt is a database of proteins. So you can look there for any protein, and you have a very detailed description of that protein. So I have focused here on the gene ontology annotations, but you have a very, very rich description of this protein and every protein in general. Then we have RNA, but there are many different types of RNA. So we have already seen messenger RNA, that is the standard one. But then you have non-coding RNA. And inside non-coding RNA, you have housekeeping non-coding RNA that, for instance, transfer RNA that is used to construct the proteins by the ribosome, or ribosomal RNA. So this is one kind. And then you have the regulatory non-coding RNA. We have already seen the microRNA, but it is not the only one. So you have long non-coding RNA, a small nuclear RNA. So you have many different kinds of RNA. And all these ones, somehow they control the transcription or the translation of the DNA into RNA or RNA into proteins. Then here we have different roles of these RNAs. So you have riboswitches. So somehow this riboswitch, depending on some metabolite, allows the expression or disallows the expression of a gene. Then you have, for instance, RNAs that help to pack some RNA inside the virus, or ribozymes that are enzymes made of RNA. So you have many different roles of all these RNA, and it controls, but they participate in many processes of the cell. Then the RNA itself, it has some structure. So it must have some three-dimensional structure. So this is how this particular ribozyme looks like. And typically, another way to represent, so this is three-dimensional structure. So this is how it really looks in real life. This is a representation somehow of the structure here. So very often you have this pairing because the two strands are complementary to each other, so they form a strand. But sometimes the strand opens like here, so you have loops, so you have a mutation, and then you have a small distortion of the strand, and the loops can be larger or smaller. So this is the typical representation of the RNA structure. And you have different pieces of patterns in this structure. But at the end, everything has to be three-dimensional. So at the end, you have atoms placed in different positions in space. And then how these the genes are translated into or transcribed into RNA. How is that? So there is the RNA polymerase. So somehow the RNA polymerase has to transcribe this gene, but there is a region before the gene that it has to bind before transcribing. This region is called the promoter, and the promoters can interact with transcription factors. So sometimes, very often, you need some transcription factors, which are proteins that bind to the RNA polymerase in this position, so that once all of them are bound, then the gene is transcribed. And the binding of these transcription factors can be also regulated by these enhancers, which can be regions of the DNA that are relatively far away, and that somehow they are connected to the transcription factors through activators or whatever. So you see that the expression of a gene is not that simple. So you need all these other actors to participate so that they somehow regulate how much gene is transcribed in a particular situation. And here you have an example. So let's say this is a famous sequence of genes, this LAC-Z, LAC-Y, LAC-A, that they are used to process lactose. So bacteria in general, they prefer, cells in general, they prefer glucose to function. But if there is no glucose, then, and there is lactose around, then you can express these genes to take benefit, to have benefit of the lactose available, to use the lactose available. And then let's say that there is no glucose and that there is lactose. And then somehow, so these are the promoting regions, and then there is a protein, this CAT, that binds to this. There are three places where they can bind. And then if this protein is bound, then the RNA polymerase can express these genes. But if there is a lot of glucose, then there is another protein that binds here, and then when it binds here, it prevents the RNA polymerase to bind, so that this set of genes cannot be expressed. Then you have all possible combinations. So there is no glucose, but also no lactose, or there is a lot of glucose and lactose, and then depending on how you regulate these, how proteins bind to these regions, these genes will be expressed or not. Then another way to regulate is through phosphorylation. So you have these transcription factors that we said they are the proteins that bind to the promoters and these enhancers. And then simply by phosphorylating those proteins, you can regulate whether this transcription factor is active or not, or how much active, and so on. So another level of regulation. So you see that gene expression is something that is extremely regulated. And these regulations, they also come in at different levels. So we have seen, for instance, that this is here where it was. Here we have some example. So to express these genes, you need these proteins. So somehow you need combinations of these proteins. So these proteins have to be also expressed. So that is a kind of hierarchy where the level 1 genes, maybe some transcription factors that regulate some level 2 genes that are also some transcription factors of some proteins that are participating in metabolism or whatever. So you can establish levels of who is regulating whom, and then you have a hierarchy of transcription factors. And then you have regulation motifs. So there are typical patterns on this regulation, on this gene expression. So, for instance, you have once you have a messenger RNA, this messenger RNA can promote itself. So maybe this is translated into a protein, and this protein somehow promotes the expression of this mRNA. So it is a kind of positive feedback loop. And then protein 1 also, or RNA 1 also promotes RNA 2. And then the more RNA 2 you have, it inhibits RNA 1. So here you have a positive feedback loop. Here you have a negative feedback loop. So if this one increases, this one will increase. But because this one is increasing, this one will decrease this one. Okay, so this is a negative feedback loop, and this can be another typical pattern, regulation pattern, where you have instead of 2 components, you have here 3 components. Negative regulation, negative feedback regulation is something very, very common. Then, how do you measure the gene expression? So you measure the gene expression using some wet lab tools. And these wet lab tools, they are based on different technologies. Here you have all possible technologies. I would say all of them, they are very much used depending on what is your purpose and how many genes you want to measure at the same time. So RNA sequencing is one that is mostly used. Now it is very cheap. You can have access to many genes at the same time. All the others, they are focusing in a few genes mostly. And every time I give you a paper, that paper is very useful to understand all this stuff. So if you are interested, you can follow them. Then genes are something that are expressed over time, so they are regulated over time and space. So for instance, here in all this development cell biology, so is for instance how the embryo develops over time. So typically they use flies because they are very easy to monitor, and there are not so many ethical issues with fly cells or insect cells in general. So for instance, you can look at a particular protein. So this particular protein, HB, is expressed in these red bands, and it depends on over time. So at the beginning, it is expressed here. At the end, you see that at the head of the insect, it is not so much expressed. So depending on where you are in with respect to the whole individual, so each one of these is coming from a single cell. There are many cells here, and then these cells are expressing, or these cells are expressing HB, and these cells are not. Okay? And this is fluorescence microscopy typically. Then you have this idea of differential gene expression. So let's say that I have different kind of cells, or I have different conditions, healthy and someone that is healthy, someone that has a particular disease, and I can look which are the genes that these healthy people are repressing or they are overexpressing with respect to this one. And that is actually your practice this year. So it is to identify for a particular disease, which are the genes that in healthy people, they have a different expression pattern from the diseased people. And this happens for conditions, for environmental reactions to the environment, to different cell types. So you have cell A, cell B, cell C, and then each different cell type has a different gene expression pattern. And in general, this problem of comparison is called the differential gene expression. And here you have, for instance, typical result from this kind of analysis. So here you have in this axis, you have the how many times you have more expression of one with respect to the other. So this is the fold change is so you have how much concentration of the gene in cell A, a concentration of the gene in of this, how much it is expressed. So you have this ratio, computer log, usually you do it in base 2. So let's say that the two genes are equally expressed. So this ratio will be 1, the logarithm will be 0. If they are equally expressed, this is called the fold change. Fold 3. And this is the logarithm of the fold change. So the fold change would be 1, the logarithm of the fold change would be 0. And then, depending on how many cells you have seen of each kind, you can associate a p-value. So for every cell, you have a different fold change, and then you have a p-value associated to how sure you are that the fold change is really 0. It is something else. Okay, then some genes do not change among cells. So all cells, they more or less express these genes in the same way, because these genes are very, very basic to life, and they are related typically to metabolism, so this energy production, cell maintenance of the cell, this kind of stuff, they are very much conserved in all cell types. So these genes, the expression of these genes help you to normalize between experiments. So every time you run each one of these techniques, the results can be quite variable. But because you know that these genes do not change, then you can use the expression of these genes to normalize and to somehow compare results from the rest of the genes that you are interested in. Then let's say that I know that there are some genes that are overexpressed or repressed. So I know which genes they are. They are these genes here that I have marked. And then I need to understand them. And to understand them, somehow I need to correlate them with phenotype. So for instance, the phenotype can be how much sugar, how much glucose they are uptaking, or how much fluorescence of whatever other gene is I have there. So there are ways to correlate your gene expression to the phenotype that you are interested in. And these random walks, I don't want to enter into the details of these analysis tools, but if you are interested, you can look for them. And so you can relate to phenotypes, but you can also relate to biological process through these gene ontology terms. So now you can look, okay, all these genes, which are the biological processes that they are associated with. and then you have an enrichment of these biological processes. Same for pathways. So here they are signaling pathways, and KF is a well-known database of pathways. And then also, what do I want to say here? So typical workflow, I guess. So first you start with this differential expression analysis, then you do some statistics, you identify which are the ones, the genes that are differentially expressed, and then you try to search these genes into different databases to understand what is happening there. So what is the difference? And these genes, what are the pathways, which are the processes, which are the regions of the cell that are somehow affected by these genes. And you can have these differentially expressed genes, which are called biomarkers. So let's say that you have a person with a given disease, a person without that disease, and you have identified that protein B is different between these two conditions. And then you can use, this is called a biomarker, and then this biomarker can be used for different purposes. So you can use it to diagnose a disease, and you can use it to select a therapy. So for instance, some people, not all people having this disease respond in the same way to a given therapy. So you can try to classify the patients according to how they will respond, and you use the biomarkers as a predictor of that response, or how the disease will evolve over time. So you can also use these biomarkers for that purpose. You can use non-genomic biomarkers. So genes are very important biomarkers, but we have seen that all these genes, they also, you have a lot of regulation. So you can use microRNAs or RNAs of different forms, or methylation states, or what else do we have here? Yeah, methylation. So you can use many other things that that somehow regulate the gene expression to use them as biomarkers of a particular condition. And then here I'm listing different databases. These ones are the ones that maybe you want to use to identify genes that are related to a particular disease. So all of them are very well known. Gene expression omnibus, array express, European. This one I don't know so much. This one, but all of them are pretty much very well known. And then I'm putting here software that is somehow used for that. This is also very well known. Captive, Lima. And then for the analysis of the enrichment, so terms, enrichment is every time that you construct a list of genes, you will have a lot of annotations. And this lot of annotations, because you have seen already for HER2. So you have many labels, and just by chance, you will have many labels occurring. How can I detect this an enrichment of labels? So some function is especially represented if I take randomly. So let's say that there is no signal, no pathway is affected, or no function is affected. Then I would have the labels distributed by chance. And then can I detect labels that are somehow clustered? They don't occur by chance. They occur much more frequently than they would occur by chance. And this is this enrichment analysis. And the same for functions, the same for pathways. So, and this is software or databases that help you to do that. And then you can go one level up. So these are metagenomics. So metagenomics is, let's say, so we have talked of DNA, RNA of a single species, but what if I have a microbiome, for instance? So I have many bacteria of many different kinds, and now I can break all these bacteria. I will not identify each one of them independently. So I break all the bacteria and I analyze all the DNA that I have in that soup. And then this is called metagenomics, and it is a whole field of research. So it is not analyzing individual species around, but collectivities of bacteria, typically bacteria. And then another field of research is this phylogenomics. Phylogenomics, it appeared a little bit at the beginning with the viruses. That is how the different genomes are related. So I have an ancestor. This ancestor starts evolving, and at some time it is very different from the original ancestor. Okay? And maybe two populations, they evolve in different ways. And then I can try to make a kind of tree, trying to say which ones were the ancestors and how they evolved. The way to do that is you start analyzing what you have accessible, that is the leaves. So these are the current species. And then the current species you see, so let's say that you have two species, which is the minimum number of mutations that I need to do to make one into the other. And that somehow gives you a distance, a genomic distance between two genomes. Any question up to here? No? Yeah.
I didn't understand the gene enrichment.
The
Gene enrichment.
gene enrichment. Yeah. So let's say that you have these just by chance. Sometimes you find genes that seem to be differentially expressed between these two conditions. So let's say that you have 5 disease people, 5 healthy people. And in these 5 and 5, you find a lot of genes that are differentially expressed. But some of them are simply just by chance. So the labels associated to these by chance genes labels, gene ontology labels. So these are that we have that in this kind of ones here, this kind of labels. So each gene has a number of labels associated. So because they are just by chance, these labels will also be distributed just by chance. Okay, so they will be randomly distributed in these 1 million possible molecular functions. But so you expect that. And now let's say that this disease is truly affecting, I don't know, this function. It is affecting this function. So on top of the by chance distribution or the random distribution, you would expect that there is many more associated to this function, because this function is really affected by that disease. Okay, so this gene enrichment or label enrichment is, can I detect this clustering of labels in one particular function on top of the level of noise that I have? No? Maybe later during the break.
Okay.
Okay. Any other question? No? Okay, so then let's continue a bit more. So next lecture is on protein systems. So protein system, this is a drawing, but it somehow tries to represent how the cell is or how packed the different components of the cells are inside the cell. So here you would have, this is a myelinated axon in the central nervous system. So you have here a lot of microtubules, then you have here actin, then you have here the some membrane, some pores in the membrane, and you see that everything is free of the fact. This would be probably the nuclear cell, and so it is really packed. And something also that you see is that, for instance, these pores, they have a particular shape, and this particular shape is very important for its function. So many of the important molecules in the cell, most important ones, we would say, are proteins. So proteins, they are made of amino acids. This is how an amino acid looks like. So it has a backbone, or it has a part that is always the same. That is, all these atoms are always the same. And then there is a region that is variable. And then this region that is variable, so the different amino acids, they bind through this carboxyl group and this amine group. So these three atoms come together, and then there is a free bond here and a free bond here that bind each other, and then they this is called the peptide bond, and then you take some water out. So these three atoms will make some water, and then you have the peptide bond here. And now you have two amino acids together, and you can continue this into a longer chain. And yeah, as we said, all the amino acids, they have a fixed pattern. So this is the part that is fixed, always this one. And then depending on what is the side chain, you have different amino acids. So you have arginine, histidine, lysine, and so you have different substitutions there. And then, depending on what you substitute, you will have different properties. So for instance, all these substitutions, they have a positive charge. And this one is not represented, but it is also positively charged. And these ones, for instance, they have a negative charge. And then these ones, they are polar, and so polar means they don't mind being in water, and they are uncharged. And then you have special in some way amino acids, and special, for instance, cysteines, they tend to bind each other. So when you have a cysteine and another cysteine, they may make a contact with them. And then you have amino acids that are hydrophobic, so they are uncharged and they don't like being in water. So you try to be at the core of the protein. This tends to be in the outside of the protein. And you can classify them according to different properties. So whether they are small or very small or large or polar or charged or hydrophobic, aromatic. So you can classify them in many different ways. There's a standard code for amino acids. So this is a standard way to represent amino acids with a single letter. And then these chains of amino acids, they have a structure. So this structure, just the sequence is called the primary structure. Then this sequence tends to form, there are a couple of patterns that are very much repeated. One is making a helix, that is called an alpha helix, and the other one is having two parallel strands, that is called a beta sheet. And then these helices and beta sheets, and there's another one that is not represented here, that is a loop. And then these secondary structures, they tend to organize themselves into space. Something called tertiary structure. This is the shape in 3D. And then you have two domains or two proteins that bind each other, and then they form a complex, and the complexes are called the quaternary structure. So then there are a few forces that make this happen. So why you have alpha helices, how these have, why these alpha helices and beta sheets make a 3D shape in space. And these are some of the forces that are there. So sometimes you have van der Waals interactions. I have another picture later, which is more clear. Hydrogen bonds, then you have ionic interactions between positive and negative charges, and covalent bonds. So these are, for instance, two systems that are bound to the sulfurs. And the beta sheet, you can have different ways. So you can have, for instance, this anti-parallel or parallel or whatever. So you can have several of them. You can also have this kind of structure. So you have a beta sheet, and then an alpha helix, and then another beta sheet. And here, the connectors here, they are the loops. And this is what I meant before. So for instance, these van der Waals forces, you have two atoms that are in principle, two molecules that are somehow apolar. So they don't have any polarity here. But when they get close to each other, somehow they induce a small dipolar moment. So somehow the negative charges are shifted and attracted by this positive charge, and the same here. So something that is in principle neutral becomes a dipole because of this shift of charges, and these are these van der Waals forces. Then you have salt bridges. For instance, here you have a positive charge, a negative charge here, and then they somehow tend to attract each other. And hydrogen may be involved in this attraction, and then they are called hydrogen bonds. And then you have hydrophobic regions. Hydrophobic regions, they tend to avoid being exposed to water, so they tend to be at the core of the protein. And then all the hydrophilic regions, that are the blue ones, tend to be in the outside. Then you have these disulfide bonds between cysteines. You have maybe you have a metal, and this metal somehow induces the shape of the nearby atoms, how they are arranged with respect to this metal. And then once you have the 3D structure, you can analyze locally the different properties. So how much hydrophobic you are. So do you like being exposed to water or not? And then you can put a number on that. Or what is the local charge? So some amino acids that are negatively, positively charged, negatively charged, and you can analyze locally the properties. Any question here? ¿Una cuestión yo que llevo? Okay, so we have seen that proteins have locally different properties, uniformity, charge, and these properties, they may also change with some environmental conditions. So for instance, the pH, so the pH controls what is the local charge, and you can see how at different pHs, the different amino acids can release a proton or accept a proton. And also the different salt conditions around they may change the structure of the protein even. So depending on what is the salt and what is the salt concentration, you have the same protein that is folded like this, like this, like this. So the salt concentration is also something that is changing a lot the shape of proteins and the pH. Those are the two main conditions. And there are proteins that are intrinsically disordered. These are these IDPs. I don't remember the exact number, but it is something like 30 or 40% of the proteins of the human proteins, they are intrinsically disordered. So they look like a mesh there. And on the other extreme, you have proteins that are very much folded. And then some, you have things in between. Some domains or some regions are folded and some others are not. And actually, it could be even the same protein. So the same protein, depending on what is the environment and depending on who is acting on it, so with whom it is interacting, it may be in this state or it may be in this state. So you have a whole range of possibilities. So the same protein in all possible states, different proteins, some of them with in this state, some others in this state, or yeah. So it is a mess. And let's say that it is a protein that likes to fold. So it has some particular folding, but this particular folding is not fixed. Actually, proteins need to be flexible to perform a function. And we will see it later that they need to move. And at different conformations, so at different shapes, you can imagine that for instance, the electrostatic interaction of the protein, of the different protein regions, or the different protein regions with the environment. So they are different. And if they are different, at the end, you can put a number of energy. So you can put something that is called the energy landscape. So the energy landscape is, depending on what is the shape, somehow this is representing the shape. Always, these proteins have thousands of atoms, but you have to look at something discrete or something that you can manage, because otherwise you have thousands of atoms. So let's say that you have 1,000 atoms. Right. Let's say that you have 1,000 atoms. So if I have 1,000 atoms and each atom has 3 positions in space, then I have 3,000 variables. This is X of the first atom, Y of the first atom, Z of the first atom, X of the second atom, Y, Z, all the way to the 3,000 variables. And for each one of these combinations, I have an energy. I have an energy for each configuration. I have a different energy value. So I have a function that is too complex to look. My energy function is too complicated. So let's try to simplify it so that I can at least plot it. So let's say that somehow you are looking at what is the angle here and what is the distance between 2 domains or whatever. So for every configuration you have a different energy, and some of them they have very low energy, and some of them they have very high energy or much higher energy. And then that means that if, for instance, you have this configuration here, this conformation, it will tend to the minimum energy state. So if it is here, if it is like this, then let's say that the minimum energy is closed, then it will close. Okay? And if you want to go back to the open states, you need to add some energy. So this ATP is typically used to do that. So you have it in this low energy state, then you use ATP to go back to a high energy state. And so folding and protein dynamics is controlled by these energy landscapes. And also you may look at the protein structure from this perspective. So when it binds to something, and here we are showing binding to a substrate. So when it binds to something, it might be that the shape of the protein doesn't change. So the active site, the region that is interacting with the substrate is the same before and after binding, but it may be that it changes. So the substrate interacts with the atoms in the enzyme, for instance, to change the shape of the active site. Here you have also a representation of a protein interacting with a ligand, and whether this ligand is able to change the protein in some way or not depends on the protein itself. And everything at the end is interaction between atoms, and these electrophoretic interactions and all these that we have seen before, van der Waals, bonds between sulfides or I don't know, hydrophobic interactions or whatever. So these kind of forces are the ones that control the energy and control the shape of the protein. Also, there are a couple of interesting ideas. One is that substrate interact with the protein at the active site, but the shape of the active site can be controlled by another place that is called the allosteric site. And then you can, by controlling the shape or binding something to the allosteric site, then the active site changes the shape, and then it cannot bind the substrate anymore, or you allow the binding. So before the allosteric site activation, the active site is not active because it doesn't have the right shape. So the interesting idea is that places that are away from the active site may control what happens at the active site. Then this idea has already here, the protein folding is controlled by an energy landscape. So some of the states, so let's say that this is the minimum energy state, and this is how the protein is able to work. And then you have all kind of intermediate states that somehow it is partially fold. And then there are enzymes that help to fold. So these chaperones somehow help you. Let's say that you are in this partially folded state and you need a lot of energy to go to here. And then the chaperones help you to overcome this energy barrier. And also you might have misfolding states. So for instance, amyloids that are very important in Alzheimer's is because the proteins take a shape that is a very long fibrils, very long fibers. And these very long fibers, actually, they are not functional. So they shouldn't be there. They should be folded in some other way, but somehow they are misfolded and you don't have the tools to break these and take back the amyloids into the correct shape. Then you have the structure of the protein, and on top of that, you have a post-translational modification. So you have the sequence of amino acids, and you may change that sequence somehow, decorating it with different things. So you can decorate with methyl groups or acetyl groups or these bar groups, or you may cut the protein at some particular location. So the final protein that is active is the one that is cut, not the original one. Or yeah, there are all kinds of modifications. Phosphorylation is one of the most common. All kinases means phosphorylating. Then, depending on the decoration that you put, so all these tags somehow also control the fate of the protein. So there are some proteins that have to go to the cell membrane, but they are somehow manufactured here in the plasmatic reticulum by the ribosomes. How do you send that protein to the cell membrane? So there is by these post-translational modifications, somehow they encode what is the fate, where they should go, and then you make some post-translational modifications, and then it says this protein has to go to the cell membrane, and it is encapsulated. Here it is going the other way around, but somehow here you can say this one has to go there. Then they are put into vesicles, and these vesicles go to the cell membrane, and the same for all other locations in the cell. And these mechanisms work going out, but they also work going inside. So you can take proteins from the exterior, put them into vesicles, and these vesicles will go to different places depending on the modifications that they have. Then the level of proteins that you have is extremely important. So for every condition, you need a certain amount of proteins, and having too much would cause an overreaction, and having too few an underreaction. So you need the right reaction to the current conditions, and all this is called proteostasis, and proteostasis is related to absolutely all biological processes. So you need the right amount of protein, and to do that, you control the protein level. So there is a mechanism for the proteasome that destroys the proteins that are not needed anymore, and somehow you through the proteolysis and the protein creation by the ribosomes, you control how much protein you have, and then you have also the ribosome is protein synthesis, and and these chaperones, we have the chaperones here. So the chaperones also help to control how much active protein you have. So we saw that there is the regulation to mRNA is very much regulated, and the protein level is also very much regulated. So there are many places where you can regulate, and all of them are regulated in reality. Then, this hemoglobin is a very famous example of protein that has an ion inside, and then this ion somehow controls the shape of the nearby atoms. And actually, this the one this hemo group is the one that transports oxygen. So the red cell is full of these hemoglobin molecules, and that is why they can transport oxygen. Then proteins is not the only thing that we have. We have proteins in combination with other things. So, for instance, we have lipoproteins. So lipoproteins are combinations of proteins and lipids, and then these combinations can be relatively small, larger, even larger, and then the lipid and the protein composition is different in each case. So, and there are many different kinds. So these are kind of big families, big groups of structures that we can have. So you have a mixture of proteins with phospholipids, with cholesterol, with triglycerol, with whatever, and in different proportions. So, and you can imagine that all this is very valuable. Then also we have peptide glycans. So peptide glycans is a mixture between proteins and sugars. So you can have a part that is protein and another part that is a sugar. And this can be used by typically the bacteria. The bacteria, they use these peptide glycans to it is part of the cell structures or of the membrane structures. For instance, this gram-negative, they have a peptide glycan here between the two membranes. And as we said before, proteins are something that this is a PDF. So proteins need to move, and they need to be flexible, because otherwise they cannot perform their function. So this was a video of the life cycle of the SARS-CoV-2 virus. This was just to illustrate how infections took place. So these are our cells. You have the virus. You see that the virus, they are flexible structure. And actually these green points here, the green points are sugars that they are used by the virus to escape the immune system. So the immune system is very good at recognizing foreign proteins. So this is a foreign protein. This whole thing is a foreign protein. But if you cover it with sugar, then it cannot see the protein, because the antibodies, they cannot attach to the protein. This one, it was presenting the structure, but the one that I want, they are here. Yeah. So they need to recognize the receptor of your cells. And then once they are recognized, you see that the proteins need to move and to change, and so that they are flexible objects. Otherwise, they cannot perform their function. The protein is not something fixed. You can watch the video yourself. So, and how to determine the structure of a protein? So either use X-ray crystallography or NMR or cryo-electron microscopy. Actually, what we do at the lab is this thing, cryo-electron microscopy. And now you have these computer predictions of structures, of protein structures. AlphaFold was the most famous one, and probably it is still one of the best, or if not one of the best. But now it is open source, but for a long time it was not open source. It was the Nobel Prize of this year, no? Physics or chemistry Nobel Prize of this year. And the input to predict the structure is proteins. So a species, so humans have this protein, but this protein is shared with many other animals and with small modifications. And then what they use is this multiple sequence alignment between the human proteins and the same protein in the different animals. And then they analyze which parts are conserved, which parts are not conserved. And from these so conserved regions, probably they have a very fixed structure, because otherwise, if we have seen the function depends on the structure. So if something is not important for the structure, probably it can change a lot in the genome. So between different species, regions that are very much conserved, they must have a relatively fixed structure. And then somehow with neural networks, they are able to predict the location of each one of the atoms. So what they are predicting is this thing that we mentioned before. Okay, so then, these are more and more videos showing that proteins need to move to be able to perform their functions. So substrate, enzymes. Yeah, so you see that everything is moving. And for instance, enzymes typically what they do is that they reorient the substrate so that they can bind in a suitable way. Because otherwise, just by chance, it would be very difficult that they find the right orientation. And then the protein around. So the protein around helps the two molecules to bind in the right orientation. You see that they need to move. Otherwise, they cannot perform their function. Okay, so, and this applies to enzymes, they apply to transporters through membranes. This you can see yourself. They're very beautiful videos about these things. They also apply to motor proteins or flagella that bacteria use to move. It is an impressive. It's an impressive. This one, let's see. See now. Okay. So it is an impressive machine. Actually, these proteins, they're called macromolecular machines. So macromolecular machines, it is like a rotor. This one is like a rotor. And every one of these is a protein. All of them are proteins. This is just showing how they work, but this is actually so and this is interesting. So if something is moving, it needs energy to move. So where is this source of movement? The source of this source of energy. The source of energy typically is a in this case it is a gradient of protons. So there are many more protons outside than inside, and this gradient of protons creates the energy to move. Okay, then antibodies, everything is controlled by this. And then proteomics. So we said we have genes. These genes are translated into transcripts and transcripts into proteins. We have seen that these are alternative splicing and post translational modifications. So we have around, let's say, 20,000 genes, 100,000 different transcripts, and about 1 million different proteins, because they can be in different states and different post-translational modifications states. And then you may wonder, you may want to measure how much is the concentration of a particular gene, or not a particular gene, a particular protein. So it is looking at the end of the process, rather than this RNA-seq. The RNA-seq is looking here, but how do we look at there? And then there are a number of tools. The most, or one of the most powerful ones is this mass spectroscopy. So what they do is they take the proteins, they carry it into small pieces, and then somehow they are make very accurate measurements of what is the mass of these pieces. And then with this accurate measurement, they are able to assign it to a peptide. So they say, oh, that this 4 amino acids work together, and they identify very accurately which 4 amino acids. And then they look at the different proteins that you have, and they find these 4 amino acids where they are in your proteins. And then again, it will. So it is like having very short reads. So very short reads. So you have the full protein, and then you have the pieces. You have many different proteins. So say I will do it with 2, and then you have pieces of this one, this piece, this piece, this piece, this piece, this piece, and here you have only amount. And then what is and you have pieces everywhere. So at the end, you recognize this was the protein that this person had, and you do it at the kind of not protein by protein, the bulk of cells. And that is also now single cell proteomics, which is very new, but still it is you have many proteins in there, and there are many other tools to do that. So electrophoresis, ELISA. This is surface plasmon resonance, CELDI. So there are many different tools to measure how much protein of a given kind you have. Another thing is that these proteomics also, or the proteins also change over time. So you have different proteins expressed in different levels, in different places, and different times. So that is another range of tools, this fluorescence microscopy, where you can target the protein. So for instance, you tag these isotones with cyclin, and then you tag the whatever, and then you can see that protein. So there is a lot of that protein I have tagged in here. There is nothing here, and there is a little bit there. So you can locate over time where these proteins are, and you tag with a marker. These markers are these fluorophores, for instance. One of the best known is this green fluorescence protein. So the green fluorescence protein is something that resonates when you illuminate with some particular laser. It can absorb the photons, emit the photons at a different wavelength. And this green fluorescent protein, somehow you have engineered the cell so that when this protein is expressed, so when this protein is transcribed and produced, it also produces this one that is attached. So somehow you have linked these two. And then if you have linked the two, you can you don't see your protein. What you see is the green fluorescence protein. And there are many markers like this. Okay, something else is protein-protein interactions. So when two proteins interact, this we have called the quaternary structure. Then the same kind of force is applied. So these hydrophobic, hydrophilic bonds, salts, or whatever, they also apply between two proteins. So now I can try to study these protein-protein interactions. There are a number of databases and ways. These are technical ways of measuring protein-protein interactions. And databases storing this data. And for instance here, and these protein-protein interactions. So for instance, this is a protein that has some catalytic activity. And the catalytic activities, they are also classified in some kind of closed dictionaries. Because otherwise, they would be, if you allow anyone to describe it in any way, it would be too messy. So these ontologies, and this is an enzyme ontology, they are meant to limit the spread of the different, to make you use words from a dictionary, because otherwise it would be too messy. So when you look at what is 2710.1 in that dictionary is something that catalyzes this reaction. So you see that everything tends to be very organized in this database. And then ways to describe that we have seen already in ontology, these enzyme collision numbers, and yeah, there are many others. All of them, they are very, very famous. Then the function of most proteins is unknown. So you know the function of some proteins, but many of other, you don't know what is the function. And then there are a lot of algorithms that they try to predict what the function will be. And they will use different properties. So for instance, they may be based just on the sequence. So if two proteins have the same sequence, probably, and I know the functions of one, probably the functions of the second will be similar. Or I can do the same reasoning with the structure, or if a protein interacts with this protein and this other protein also interacts with this one, probably these two have some shared function. And so all kind of reasoning allow you to infer what is the function of the protein. Then these protein interactions, they are structured into pathways. So you have, for instance, here different pathways for this is human Homo sapiens pathways, and then the pathways look like this. These are like what we saw in the first lectures. So there are graphs that represent how the proteins interact with each other. So this protein somehow interacts with this, but this one inhibits this interaction. And they all go at the end to these protein 3D structures that have to interact with each other. And databases of pathways in this case. Yeah, that was all. Any question? So you see that this is systems biology. So systems biology is we have a set of agents. Agents here have been proteins and genes, and somehow they interact with each other and they regulate each other, and the level of each one is really regulated and adapted to the conditions that cell is having. Any other thing? No? Venga, pues vamos.